Hey Beaklings, so in this video we're going to go ahead and review oxidative phosphorylation, which is the electron transport chain and chemiosmosis. And like I said for the other video where we reviewed glycolysis and the Krebs cycle, this shouldn't be where you're learning this stuff, right? Hopefully you already drew the pictures with me and you heard my explanations of how this stuff happens, and hopefully you already watched some videos and read the textbook and things like that. But once you feel like you understand this should be a breeze and just a nice quick review for you. Okay, so here we go. So we're definitely inside of our mitochondrion here, and as you can see in the picture, the majority of oxidative phosphorylation happens right along this inner mitochondrial membrane, right? So we have the electron transport chain and chemiosmosis in this final step of cellular respiration. And if you notice, we have all these twists and folds to this membrane, and that really increases the surface area, right? So the more twists and folds of this inner membrane, the more of these protein complexes we can have to do more oxidative phosphorylation. Right? And at the end of the chain, you know that we're going to have oxygen, okay, which is O2, diatomic gas molecule. We're going to have half of that reacting with some electrons as they oxidize the end of the chain here at this protein complex, pull the electrons off, and add on some hydrogen ions or protons to create water. Okay, so that's the electron transport chain. Now, what was the whole point of this? Well, let's see. The spatial arrangement of electron carriers, okay, make it possible to use the energy of electrons to create a gradient. Remember, we said this is step by step by step, okay? So as NADH drops off the electrons here, it's actually used to turn this protein complex on, and it's a pump. It's a hydrogen ion pump. So pump, 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 pump. You're pumping hydrogen ions up. FADH2, he delivers over here to a second protein complex. Okay, this picture is very simplified. I'll show you a more complex one at the end. But the electrons from FADH2 get delivered a little further along in the chain, and thus this protein is pump, 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 pump. But as the electrons keep getting passed, okay, and it's not exactly like downhill like this, they can kind of move around. But point is, as they go downhill, okay, you lost your energy, so maybe by the end of it, this protein pump here would be, or this proton pump would say, pump, pump, pump. So he's not pumping as much, and by the time the electrons reach the bottom, they're low energy, and who better than the very electronegative oxygen to sit here and say, hey, sure, I'll take these electrons, and he takes some protons as well to form a waste product, in this case, water. Okay, now what was the whole point? You ate your food so you can oxidize it, right? And by oxidizing it, we already got some ATP, but we mainly got high energy electrons from glycolysis, the grooming of pyruvate, and now the citric acid cycle. So what do we do with these electrons? We turn the energy of your food into a, whoa, it's like a proton motive force, right? It's a hydrogen ion gradient up here, and that's a lot of potential energy. So what happens is the energy of this gradient will now facilitately diffuse. So this was active transport, right? We went from an area of low concentration, there's not too much hydrogen down here, to an area of high concentration, right? And what happens now is, well, that potential energy can facilitately diffuse back down through ATP synthase, and when that happens, this guy starts to spin around and around and around and around, and thus it actually opens up the active sites on the bottom. Down here there's like bulbs, and when the active sites open up, ADP and P fit in there, inorganic phosphate will jump onto adenosine diphosphate, and you're going to make a whole lot of ATP. Okay, so that potential energy is what we use to go ahead and phosphorylate ADP, right? So again, it's roughly 28 ATP, so we got about 34% of glucose's total energy doing this process, and that means that you're not even as efficient as like a pickup truck driving down the road, but that's okay because a lot of the heat that's released, don't forget, through thermodynamics, a lot of the energy transfer that happens here leads to an increase in entropy, right, or random heat energy. And, 34% of glucose's total energy is good enough for us, right? So as long as you're breathing oxygen and eating food, you're going to be able to get a bunch of ATP out of your molecule. Yes, you're going to lose heat in the process, but we've evolved to need to be about 98.6, right, or thereabouts. So point is, pretty good process, not necessarily the most efficient process, but now it's what we need it to be, right? All right. And the final step is called oxidative phosphorylation. Well, why is that? Because, think about it. The energy gathered from oxidation reduction reactions of the electron transport chain is used to phosphorylate ADP to ATP, right? So you get oxidative phosphorylation. Now, what effect would an absence of oxygen have on this process? Okay, now here's the answer that a textbook I used to use gives exactly, right? Without O2, the electrons wouldn't be pulled down the electron staircase. Okay, I don't know, and then of course no ATP would be generated there. I don't know if I would say they get pulled down. Okay, the oxygen doesn't necessarily keep this chain yanking these electrons, right? It basically, what it does is it takes the electrons from the very bottom of the chain, right? So oxygen in this case gets reduced. It oxidizes, okay, this complex, and thus gets reduced so that once you join with some protons, you can form water. But it's not like it pulls the electrons down like that. No, he's a very electronegative bully, but he's just interacting right here at the bottom. Okay, and here's a quick little review video for you about the overall process.
Most of the energy harvested from organic molecules during glycolysis and the Krebs cycle is stored in NADH. NADH and FADH2 molecules give up their high-energy electrons in the third phase of cellular respiration, electron transport and ATP synthase, where most of the cell's ATP fuel is produced. The electron transport chain is an array of molecules, mostly proteins, built into the inner membrane of the mitochondrion. NADH gives up its high-energy electrons to the first complex in the electron transport chain. The electrons move from one member of the chain to the next, giving up their energy as they are pulled from NADH toward oxygen. The energy given up by the flow of electrons is used to pump hydrogen ions from the mitochondrial matrix into the intermembrane space. Oxygen captures the electrons in the very last step in electron transport. The last complex adds a pair of electrons to an oxygen atom and 2 hydrogen ions, forming water. The electron transport chain has used the energy of moving electrons to pump hydrogen ions into the intermembrane space. This buildup of hydrogen ions, like water behind a dam, stores the potential energy that was originally in the bonds of glucose molecules. The backed-up hydrogen ions give up their energy when they diffuse through a special protein complex in the membrane called ATP synthase. As hydrogen ions flow down their concentration gradient, ATP synthase captures their energy to make ATP. Under normal conditions, almost all the ATP produced in the process of cellular respiration is manufactured by the energy that electron transport provides to ATP synthase, about 34 ATPs for every glucose consumed.
Okay, and like we said, the numbers aren't exactly important here because, you know, the current book that we're all using says 28 ATP here. This video says 34 ATP here. And scientists still are researching that, and they're studying it, you know, because it's important to know how efficient we actually are. Okay, and some of it has to do with how much energy it takes to shuttle the NADH and how much actually can you pump from the electrons from one NADH molecule or one FADH2 molecule, depending on where they're delivered and all that stuff. So pretty cool stuff. And hopefully that was a little bit helpful. Okay, and I just wanted to show you this very quickly just so you can see here. It's a pretty impressive process, right? So scientists had to really dive in and learn all about these different proteins and non-proteins that work together to have the electron transport chain work correctly. And a lot of these things are made from gene expression, right? Through the DNA of the mitochondrion, right? So this isn't even our DNA, our nuclear DNA, that knows how to make this stuff. It's actually the DNA that's already in the mitochondria. So this is a really cool process, and it's a lot more complex than we let on this chapter, right? So if you stick with biology and you keep going into AP Bio or eventually college bio, you're going to really dive into the electron transport chain and chemiosmosis. It's really cool stuff, okay? Just have one last thing I want to talk to you about. Okay, just going a little bit deeper here. Let's take a look. If I was to ask you a question like, well, why are there always going to be more hydrogen ions on the top of that membrane? And maybe I shouldn't say at the top. Why are there always going to be more protons in the intermembrane space, the dark gray area, than there are in the matrix? Okay, and there's a couple different reasons for that we can look at here. First of all, how many protein pumps, okay, or these proton pumps are going up? Okay, Well, we have 3 of them, right? 3 are going up and only 1, the ATP synthase, is coming back down. So you basically have 3 going up and 1 coming down. So that's one reason you should have more up top. Another reason is, well, take a look at what happens at the bottom there with oxygen, right? Oxygen is going to grab 2 protons to form water. So there's another reason that you're not going to have the too many down there, because you're pumping 3 of them up and you're stealing 2 of them all the time. However, you're not going to run out, because look what happens with NADH and FADH2. They are always going to be delivering them, right? NADH drops off 1 proton, FADH2 drops off 2 protons, right? And that ATP synthase does have protons coming back down, facilitately diffusing down through them. So you have the active transport going up and the diffusion coming back down, all right? And one other thing I wanted to talk to you about is, what about this oxygen? Okay, if you didn't have oxygen in your electron transport chain, of course the chain stops. Okay, because you're going to stay in the reduced state, if that makes sense to you, right? Because the electrons are going to get stuck, if you want to think of it that way, right, in the chain. You can't just keep feeding it, right? That first protein complex can't take NADH's electrons, right? It can't oxidize it if it already has electrons backed up in the chain. So what's going to happen then is the electron transport chain will shut down. Now, this is going to go into our next topic a little bit, but think about this. If the electron transport chain shuts down because of lack of oxygen, which should make sense to everybody, right? Oxygen is a very important part of this. That's directly affected by oxygen. Would the citric acid cycle shut down? And the grooming of pyruvate, right? Is the mitochondria kind of shutting down because of a lack of oxygen? And you might say to yourself, well, no, because oxygen is only used in this last step. Not true, however. Think about what I just said. NADH and FADH2 would have nowhere to go without oxygen. They're just holding on to those, electrons with nowhere to go. So technically, you're not going to have any NAD plus or FAD inside the matrix, because they're all going to get reduced to form NADH and FADH2 and have nowhere to go. So technically, without oxygen, indirectly, the Krebs cycle and the grooming phase will also shut down. Now, you might say, well, what about glycolysis? We already know that doesn't need oxygen. True. Okay, so what do you think? Now, it depends on what kind of organism you're talking about here, but what do you think about you? If you don't have oxygen, well, you're dead without oxygen. But let's just say you don't have quite enough oxygen, right? Rather than shuttle that NADH that you made all the way to your mitochondrion just to sit there, right, because there's nowhere to deliver the oxygen or the electrons to anyway, what's going to happen is this really cool process called fermentation. Okay, so that's really all we have left to talk about this chapter, is fermentation and how important that is. For us, it's kind of like a backup system to get more energy as we need it when oxygen isn't quite enough to meet our energy demands. Okay, but for some organisms out there, fermentation is all they need to live. So I hope this video was helpful, and I hope oxidative phosphorylation makes a lot of sense to you. Okay, take care.